Okay, in a previous part of this chapter and discussion, we posed the question, was Mendel lucky? Well, in one sense, he was, because he chose characters that have alleles located on different chromosomes. So he didn't choose characters where two different characters were located on the same pair of homologous chromosomes. Genes located near each other on the same chromosome tend to be inherited together and do have more complex patterns of inheritance than Mendel observed. What he did choose, just sort of randomly, luckily, I guess, were characters that exhibited complete dominance of one allele over the other. And he also chose characters controlled by only two alleles, no multiple alleles. What you're going to see is that complete dominance where you have two alleles controlling a character like flower color, where one allele, the purple allele, is completely dominant over the white allele, is not in fact, maybe even most common of the inheritance patterns. And in many instances, you don't just have two versions of alleles for a given gene, there may be more than two. And we call these multiple alleles. So again, Mendel was lucky because he happened to pick genes that weren't linked. He also chose characters that exhibited complete dominance, and he didn't choose any characters that were controlled by more than two alleles. So now let's look at some of these examples of other inheritance patterns. We're going to call this the spectrum of dominance. So the ones that we've looked at already would be a complete dominance behavior or pattern of inheritance. The dominant allele determines the phenotype over a recessive allele. So for example, purple flowers versus white pea flowers. Now, we also have codominance. And codominance, both alleles are going to affect the phenotype in separate and distinguishable ways. So a good example of this is something called M and N blood group. Now, M and N blood groups are not blood type. We'll get to that in just a little while. But we do have different proteins on our red blood cells. And so there is an allele that controls an M protein and a different allele that has an N protein, producing these proteins on the cell membranes of these red blood cells. So, since we have two separate alleles, the presence of an M allele or an N allele doesn't by itself determine the phenotype. These are codominant, right? Individuals that are homozygous M have red blood cells with only these M glycoproteins on their surface. Individuals that are homozygous N have only red blood cells with N glycoproteins. But individuals that are heterozygous here and have an M allele and an N allele have red blood cells with both the M and the N glycoproteins. So again, this is an example of the inheritance pattern called codominance. Now another type of dominance is incomplete dominance. And this is where the alleles for some characters are going to fall sort of into the middle of a spectrum, where a phenotype of a hybrid represents a blending of the two parental varieties. So snapdragons are a good example of this. You can have the P generation of red snapdragons and white snapdragons. Now notice that the notation looks a little bit different here. We've got the color red, and so a true breeding red snapdragon has two alleles for red flowers, while the color white has two alleles for the white flowers. When we cross these in our F1 generation, instead of seeing one color, either red or white, we see a blend. And this is because each one of these parents contributes one half of the pair, right? So they contribute one allele. And in this case, this individual has one allele for red and one allele for white. And if you think about this, if you mix a bucket of red paint and white paint together, you end up with pink. Now, if we self-pollinate these pink ones, and we see the way that the Punnett square works out, where we have this allele here and this allele here, we take this allele and move it here, and this one Here, notice now our F2 generation in the incomplete dominance case is 1 red, 2 pink, and 1 white.